Hello students, welcome to NAC Biology. As we know that except unicellular organisms like bacteria, fungi, some algae and some multicellular plants, almost all organisms reproduce sexually where there are two parents involved and two gametes like male and female gamete fuse to form zygote which develops into new organisms, right? It is very clear. But still there is differences among the parents and offsprings. They are not identical. Especially in the case of mammals, such a sexual method of reproduction is very common. But here is one method in which mammal can reproduce asexually without gamete fusion. It is very interesting, right? And surprisingly, they can produce genetically identical copies of an individual. What is that method then? Yes, that is nothing but cloning. Cloning is a method of production of genetically identical copies of an organism by asexual method. The cloning in mammals first conducted by Ian Wilmot and his colleagues at Rosalind Institute. The first cloned mammal is female sheep named Dolly. It born in the year 5th July 1996. Cloning is carried out by using a technique called somatic cell nuclear transfer. In short form, we can call it as SCNT. In this video, we shall learn how this method implemented to get Dolly. Come, let's start. For the experiment purpose, two female sheep were taken. The first sheep, named Finn Dorset, which is white-faced, act as nuclear donor. The somatic cell was taken from its mammary gland. Somatic cell is nothing but regular body cell, not an egg cell. From this cell, only nucleus was taken. Now, they selected another female sheep called Scottish blackface. As the name itself is telling, blackface sheep. Here, the egg cell was taken from its ovary and the nucleus was removed. Now, both the cell content, that is nucleus and the egg cell, were fused using electric shock. This method is called somatic cell nuclear transfer, that is SCNT. SCNT is a laboratory strategy for creating viable embryo from body cell and egg cell. The hybrid formed begins to divide normally and developed into blastocyst. The blastocyst was implanted into the body of surrogate mother, that is sheep 2, Scottish blackface. So, Scottish blackface sheep gave birth to Dolly. Dolly's white face was one of the first signs that she was a clone, because if she was genetically related to her surrogate mother, she would have had a black face. But Dolly is exactly like sheep Finn Dorset because her DNA came from Finn Dorset mammary gland cell. Dolly led a normal life with other sheep at Rosalind Institute, but she died on 14 February 2003 because of lung disease and severe arthritis. Okay students, hope this video will be very helpful for you. And if you have any doubts, please type in the comment box. And please don't forget to subscribe to our